Internal mechanism of hormone action. Hormones stimulate or inhibit the activity of cells directly or indirectly through modulation of the actions of other chemical messengers. Some hormones, often referred to as first messengers, interact with the cell membrane to increase the production of intracellular second messengers, which are more directly responsible for activation of the cell. The second messenger is usually a cyclic nucleotide, either cyclic adenosine monophosphate, cyclic AMP, CAMP, or cyclic guanosine monophosphate, cyclic GMP, CGMP. So the first messenger, second messenger model of hormone action must now be expanded to include other messengers in the temporal sequence of hormone action. For example, some hormones require calcium for activation of cyclic nucleotide formation. In some cells, the first messenger acting through induction of an inward calcium flux may activate prostaglandin biosynthesis. The newly synthesized prostaglandin activates the intracellular production of CAMP or CGMP. These cyclic nucleotides actually mediate their actions through stimulation of cellular enzymes, kinases, which phosphorylate cell-specific proteins. These phosphorylated proteins then function as the ultimate physiological effectors in the cell. Other first messengers interact with intracellular constituents of a cell to mediate their action. Then cellular receptors and hormone function. Hormone regulate specific target tissues, not all cells in the body. One might ask what mechanism or characteristics of a cell determines whether it will be responsive to a particular hormone. Molecular components of cells called receptors provide the specificity for hormone-cell interaction. So hormones regulate the specific target tissues. Molecular component of cells, so-called receptors, provide the specificity for hormone-cell interaction. The receptor concept originated early in this century with the work of Langley. So receptors may be components of the plasma membrane, or they may be cytosolic or nuclear elements. Receptors provide the means by which hormones initially interact with cells. If each type of cell did not possess specific hormone receptors, all cells might be expected to be responded to all chemical messengers. Each time a single hormone was released, all cells in the body would respond, and this would result in uncoordinated muscle contraction and relaxation, and the uncontrolled secretion of numerous cellular products such as hormones and enzymes. Therefore, cells do not possess receptors for all hormones, but rather only a limited number of different receptors. Certain cellular elements may exhibit specific affinities for non-hormone agents, such as narcotics or other drugs. These affinities provide the basis for medicinal drug therapy. Some drugs interact with hormone receptors, which may result in activation of the cell, or if the ligand, receptor binding substance, lacks intrinsic biological activity, it may act as antagonist to block the binding of a hormone. Oxytocin and vasopressin are related in structure, and both the hormones stimulate uterine smooth muscle contraction and activate renal tubular cyclic AMP production. So uterine receptors are, however, more sensitive to oxytocin than to AAP. Thus, the renal receptors are more sensitive to AAP than oxytocin. Thus, at normal circulating concentrations, each neurophysiological hormone will only activate its appropriate target cell receptor. So, vascular smooth muscle, neurohypophyseal hormone receptors are generally less sensitive to oxytocin or vasopressin than are uterine or renal tubular cells. During severe hemorrhage, however, large quantities of vasopressin are released from the neurohypophysis. The hormone may then be able to induce vasoconstriction and serve an important role in hemostasis. Some hormones stimulate a number of tissues, which suggests that each of these diverse tissues possess receptors for the hormone. Insulin is probably the best example of a hormone with multiple actions. Insulin stimulates glucose uptake by hepatocytes, fat cells, and certain muscle cells, and interact with many other cell types. Insulin is thus able to rapidly lower extracellular glucose levels, which is one of its major functions. If more than one tissue responds to a particular hormone at one time, it might be expected that all these different physiological responses would complement the one physiological process being regulated. For example, parathormone elevates serum calcium levels by releasing Ca2 plus from bone, stimulating Ca2 plus uptake from the gut and preventing Ca2 plus loss from the kidney. Each of these individual responses is important in elevating serum Ca2 plus levels. The normal physiological levels of each hormone interact with its own specific cellular receptor. Estradiol, for example, interacts with estrogen receptors, not with other steroid receptors such as those for cortisol or progesterone. Therefore, hormone receptors possess a recognition function which must then be converted into an action function. Receptors recognize differences in hormone function structure and thus provide receptor specificity. Norepinephrine and epinephrine are structurally similar, but norepinephrine preferentially interacts with certain types of adrenergic receptors, whereas epinephrine interacts more specifically with other adrenergic receptors. The pharmaceutical industry capitalizes on receptor specificity to provide hormone analogs that can be used medicinally to activate or inhibit certain tissue specific receptors.